Rappin' with ReefBum is sponsored by Bulk Reef Supply and Ecotech Marine.
Hey everybody, and welcome back to another episode of Rappin' with ReefBum. I'm your host Keith Berkelhamer. So today I have the pleasure of welcoming back Dr. Eli Meyer to the show. Eli is a coral biologist and aquarium hobbyist, and he owns AquaBiomics. AquaBiomics can analyze the microbiome of an aquarium using DNA sequencing, so they can diagnose issues and identify strategies for promoting a healthy microbiome. Eli, man, welcome back to the show.
Thanks for having me. Great to be
So
here.
before we start chatting with Eli, I want to thank the sponsors of the show, Bulk Reef Supply and Ecotech Marine. I really appreciate these companies supporting the live stream, and I also appreciate you folks tuning in. Please spread the word and hit that like button. You might even want to smash that like button so more people can find this live stream. And as always encourage the comments and questions in the chat. I'll do my best to take those and field those questions. We're going to try to have an orderly discussion here with Eli. So I don't know, Eli, it's been about 10 months since we've had you on the the live stream. Do you want to just give folks a real brief quick overview of the company and what you guys do, the types of services that you provide, just as a little refresher?
Sure. Yeah, so our company is all about DNA testing. We use DNA sequencing to identify different kinds of organisms in your tank. We started out with microbiome testing, so this focuses on the bacteria and the archaea, microbes in your tank. And over the last couple of years, we've expanded out into a new group of targets, same technology, but now we're targeting different organisms. This is our tank DNA test, and this targets everything else. Really everything that's not a bacteria or archaea is targeted by this test, and we use this to identify parasites in your tank, things that cause the major fish diseases in the hobby. Yeah, so those are our major services, both based on DNA sequencing.
So, yeah, I know we talked about the DNA sequencing the last time you're on. Are you guys kicking around even some other new services for the future, or are you just going to try to focus on what you guys are providing right now at this point in time?
Yeah, so really thinking about the development of the company in kind of three phases. And the first phase was just getting it launched with the microbiome service itself. In the second phase, we've expanded out to this tank DNA service that I described, and we've also started adding some supplements. So we're now selling live reef sand and live reef rubble. These are natural supplements that hobbyists can use to boost the microbiome in their tank. And listeners may be wondering, so what? I can buy that stuff anywhere, and that's true. There's lots of sources, at least for live sand. The distinction with our products is that we use the DNA sequencing to test them and confirm that they are free of parasites and pathogens before you put them in your tank. So it's the only live sand you can buy that comes with an ingredients list. You know exactly what's going in it. In the future, we are expanding out into, our main focus for the next steps of our service, of improving on our service, is all about the turnaround time. This has always been the kind of the major stress point for hobbyists. They've got a sick fish or dying corals. They want that answer quickly, but researchers operate DNA sequencing on a scale of months to maybe a year. That's way too slow for hobbyists. So we've been operating with a 1 month turnaround time this whole time. We've now purchased a machine that will, once we transition the service onto this new machine, this will allow us to do the sequencing in-house, and will make our turnaround time about 2 weeks. Yeah, so that's the major focus right now in terms of improving our service. Is the end product won't really look that much different for the clients, but once we have everything up and running, it should come about twice as fast. In the very long term, phase 3, we are planning a new technology. Well, actually, it's an old technology, but we're planning to use it for this purpose. We're planning a qPCR-based service. So this will be for a specific pathogen or parasite. You know, if you just want to know, do I still have ick in my tank or is it gone? This technology, qPCR, is much faster. So in principle, we'll be able to give you the answer in days rather than weeks at that point.
Well,
that's a future vision. We don't even have the instrument in the lab, but you know, it's the same technology that they use for COVID testing. So listeners may be familiar with it already. Pretty quick method. That's where we're looking in the future. Right now, we're focusing on making the sequencing-based testing go a little bit faster.
So the current turnaround time is about a month. Do you have to, I mean, does it get put into the queue and essentially does it depend in terms of when you guys receive the results back from a hobbyist? So for instance, like if somebody sends you the results in and you get it back at the beginning of the month, first, like, you know, 7 days of the month or something like that, does it kind of have to wait in the queue, so to speak, for a little while before you, you know, have to run? Yep.
Yeah, that's exactly right. Sounds like you have some experience with our service.
I do.
Yeah, it's a cycle. You know, it's about a 1 month cycle. So if your sample arrives early in the cycle, it will wait longer. So I often tell hobbyists, if you have the flexibility in your schedule, send your sample in at the end of the cycle, right before the deadline, which is typically end of the month. For a couple of years now, our deadlines have always been end of the month. So if you get your samples in towards the end of the month, you'll get your results. Your personal turnaround time will be shorter because they got there at the end of the batch. Yeah, when we get the samples in, you know, I have to accumulate enough samples in the lab in order to fill a batch. I process them. I work really hard in the lab to do my steps quickly. So sort of 3 to 4 days of full days in the lab to process all the samples. And then I send them off to the sequencing facility. Like just about every researcher out there, we're using a professional sequencing facility at a university. The instrument that your samples get sequenced on costs about $500,000. So that's certainly more than my budget, and it's more than the budget of most researchers. So we outsource it to these sequencing facilities. And I've got a good relationship with them, so they do turn it around quickly, but you know, quickly for them is a week and a half to 2 weeks. Then when I get the results, I work like mad to turn those reports out in about 24 hours. So by far the longest part of the cycle is the sample sitting at the sequencing facility. And that's why bringing the sequencer in-house is going to make such a big difference to turn
sounds like it. So what so those companies that you're sending the data out to, are they constantly tweaking their methodology to try to improve the accuracy of the results? Is that something that's always going on in that business?
No, it's funny. They're cookbook operators, you know, and I don't mean that as any criticism. It's actually intentional that everything is standardized. Every sequencing run is exactly the same. And so actually the company's name is Illumina. They're the biggest DNA sequencing company in the world, probably. They send out, you know, they sell the instrument and they also sell the reagents, the chemicals to use it. So think about it like a printer a printer company also sells the ink for their printer, right? That way they know the ink cartridge fits just perfectly in there. And it's the same way with Illumina. You know, every kit is identical, and the operators at the sequencing facility, they're following it exactly by the book. So it's the same every time on their end. They frankly don't even know what it is that's in the sample they're sequencing. They're putting it in, loading the other chemicals, and pressing the right buttons to make the data come out.
So, besides like the turnaround time, what Eli, have been your biggest challenges since you started this company in terms of you know, the AquaBiomics, you know, the services that you guys provide? What's been like the biggest challenge for you guys? What do you think?
Yeah. Well, you know, there's a lot of logistic challenges on the side of running the business that probably aren't as interesting to the listeners. But from the perspective of the technology, I'll say that one of the challenges, and this is not a challenge that's unique to AquaBiomics or unique to studying aquariums. This is a challenge for using DNA sequencing to study microbes. The way that all of us do this is we sequence the same piece of the bacterial genome. The analogy I always give people is it's like if you went into a library and just read the first page in every book and identified the book based on the content of the first page. Well, that works pretty well. And I mean, that's where the whole field of microbiome studies has come from. But it's not perfect. When you're just reading the first page, there can be cases where you can't identify that book because nobody else has ever read that first page. And so you don't have a list where you can say, well, this text corresponds to this book. Maybe my analogy is getting a little tortured here. Let's go back to DNA. So when you're sequencing just a piece of a single gene, that sequence may not be in the database. Maybe nobody else has ever encountered that particular sequence before. And so there's a chunk of the data that comes back that I can't tell you what it is. Right now, we don't talk about that in the report, just like all scientific papers also don't talk about it. But that's a challenge that some of the data we can't make sense of. And it's a challenge that will be really addressed well by moving to this new sequencing platform that we've purchased, because this allows us to sequence the entire gene. It's something like 10 times more data. Wow. Right, the whole gene is about this long, and we're sequencing this long, right? About 1/10 of the gene. So it's going to allow us to identify a larger fraction of the sequences that we get back. It'll also bring some clarity in cases where we can identify it to a certain level, but we can't identify it any further. And actually, your results have a good case in point here. Your pre-sample that we recently sequenced, that community was dominated by a family called Vibrio.
Yeah, that sort of scared me.
This is the family that includes Vibrio. Yeah, it does, right? Because we know Vibrio includes some pathogens. Well, I looked in detail into this. Your sample had so much of this group that I wondered, which types. And it turns out that almost every sequence in your sample, almost every Vibrio sequence in your sample was identical. They were all the same type. However, it's a type that we can't identify below the genus level. And that's because that same sequence is shared by multiple different Vibrio species. So again, that's a challenge inherent to DNA sequence. And sequencing a longer piece is going to help. So the reports will just get more and more informative as we start sequencing a longer piece of DNA.
So
so that's a challenge that all of us face.
Yep, I hear you. So when I had Jake Adams on a few weeks ago, he had talked about he had done some testing with you guys, and he said he was skeptical of some of the results. I can't remember if it was he had Astrina snails or if it was Aptasia. But I guess, the reports that he got back from you said he did not have this pest that he saw in his tank. Well, I just want to give you an opportunity to address that comment. And will that this new technology you're talking about in terms of 10 times more data, will that potentially resolve an issue where you're not reporting something that hobbyists might be seeing in their tank?
Yeah. So I mean, let's broadly talk about what kind of errors can occur with this technology. And that's certainly one type of error. You could have something in the tank and you don't detect it. It turns out that the test, let me step back and let me say it's not really even about the test. It's more about the sample. So for tank DNA, we're sampling DNA that's floating around in the water in your tank. That's it's a subject that in the research world, they refer to it as eDNA, meaning environmental DNA. And this is quite different from the microbiome stuff, where we're actually capturing cells and taking the DNA out of those bacterial cells, right? So in order for us to detect something in your tank, its DNA has to be abundant enough dissolved in the water of your tank for us to detect it. Now, we detect a lot of stuff dissolved in the water of your tank. We detect, fish, corals, sponges, snails, all the stuff you can see, we can detect them. But if your sample contains a whole bunch of DNA from something else, maybe it's got a lot of diatoms, maybe the sponges recently spawned and it's got a lot of sponge DNA, that happens. That reduces the number of sequences available to detect something else in the sample, right? It's like most of the sequences are taken up detecting that abundant thing. So rare species, rare members of the DNA community in your sample, we may miss them as a result. And it turns out that some of the animal pests like Aiptasia, Astorina, those don't contribute a whole lot of DNA to the water. Now, if you get me a little piece of one of those things, no problem, we can detect it. I mean, we've done this. Actually, short side note here, Astorina, these are little starfish that many of us obsessively take out of our tanks because we're convinced they eat corals. Yeah. And I believe this. Well, some of us have noticed there are black ones and white ones.
I've never seen black ones.
Okay, so several of us hobbyists in the area here, we've compared notes on this, and we're all convinced that the black ones eat corals much worse. The black ones are really the bad ones to watch out for. Well, when I go in DNA sequence, the black ones and the white ones, I find that they are in fact different DNA sequences, supporting that these are not just different color morphs. They really are different beasts, whether at a species or subspecies level. That was a tangent. My point is just to say, no problem. If you get the DNA from Astorina in my tube, we will detect it and correctly classify it as Astorina. If a sample has a very, very low amount of Astorina DNA in it, we're not going to detect it. If you're looking for something that's visible, the best, most sensitive way to detect it is always going to be visually look for Astorina in your tank. We can detect them. We detect them in lots of tanks. We detect Aiptasia in tanks. In fact, we use our Aiptasia testing, we use tank DNA testing to test the live reef rubble that we sell. And we frequently detect Aiptasia in that stuff even when we can't see it. And as a result, we can't sell that batch. So I believe that you can have a tank with Aiptasia in it where we don't detect it in the sample. That just tells me there wasn't much Aiptasia DNA in the water. But from experience, I can say that the test is sensitive enough. It often does detect Aiptasia even when you can't visually see it.
Now, what about, I know you guys also can detect like aqua-eating flatworms. Do you need like a big infestation of that, or can it just be like one infected coral perhaps where you might be able to detect the aqua-eating flatworms?
So that's another one where I'm going to say whether it's because of the prevalence in the hobby or because it doesn't contribute much to the water column, we rarely detect it. We've only detected those in a couple of tanks. I did have a colleague in Europe send me a specimen of the flatworm itself and sequenced it, and no problem. It shows up as aqua-eating flatworm. It's in the database. It sequences just fine. Yeah, so I think that, for Aiptasia and Astorina, I have enough data to say that the test is not, again, it's not the test. The samples are not terribly sensitive for Astorina and Aiptasia. If your primary concern, you the hobbyist, if your primary concern is, do I have Aiptasia, you should look. That is the most sensitive way to test for these things because you can see them. I think DNA sequencing is most appropriate for issues where you can't see it.
So what about like parasitic copepods? There's like the red bugs. I think there's been evidence that there's other types of bugs out there, black bugs, white bugs, whatever. Some people even don't like to use the term bugs because they're copepods. But how accurate can your testing be to detect those sorts of things? Because those are very hard to see with the visible eye, but you can have a total infestation, and you'll probably notice it with reduced polyp extension.
Yeah, I can tell you copepods show up very commonly on our tests. I can't think of a case where we've detected any of the parasitic copepods, but we certainly detect copepods frequently. Maybe half the tanks that we test come back with copepod DNA. So if any listeners out there have a known case of an infestation, please send me a sample. I'm happy. Let me phrase this really carefully. If it's a parasite of interest that we haven't detected frequently, and you have a sample of it that you know it's there, please send it to me and I'll sequence that thing for free.
Cool.
Because it's really useful to get that first positive result. Yeah, so I think most of the stuff that the test is really good at are the micro eukaryotes. We get excellent sensitivity for dinoflagellates, and of course there are some parasitic dinoflagellates out there. We get excellent sensitivity for ciliates, and this is another group. We know there's some bad guys in the ciliates. So I really think that is the best group of organisms to look for here. If you want to know what kind of ciliate, you want to know what kind of diatom or dinoflagellate, anything microscopic like that, this test does a really good job at picking them up.
So funny comment by Great Bearded Reef. Come get some of my Aptasia. Most have names these days like Bob, Johnny, etc. Can't wait to get another CBB. That's a copper band butterfly, I believe.
I must clarify. We have plenty of samples of Aptasia. Yeah. No need for more Aptasia.
So Planet 3D is asking a couple of questions, and I had a question basically based on what you were just saying in terms of determining all these different organisms in the tank, pathogens, what have you, pests. But going back to my example in terms of the bugs, some of the bugs are parasitic, but some are not, right? I mean, there's some copepods that will not harm acropore or what have you. I mean, is there a way for you guys to identify the good guys versus the bad guys in terms of those types of parasites?
Yeah, so it looks like this. Have to get into the weeds for a minute here. Okay,
let's get into the weeds.
When you sequence a piece of DNA rather than sequencing the whole genome, we refer to that as a genetic marker. It's one little piece that you're targeting in order to classify the organism. Okay, and genetic markers, a particular genetic marker may be really good for one group. That is, it can tell them all apart. And it may be really bad for another group. That is, they all look exactly the same for this genetic marker. The marker that we use is very bad for fish. And so people sometimes come back to me and say, well, this is strange. You told me I have cod in my aquarium and that's the only fish. No, it's not that. And I inserted a little note about that. The marker just isn't useful for fish. It detects that there's fish in your tank, but it does not correctly classify them because fish don't have enough variation in this piece of the gene. Copepods do. So this is a useful marker for differentiating copepods. It does resolve them well. So if we get the DNA from a copepod into the sample, then I have high confidence we're going to be able to identify it at least to the genus level, and really we usually identify them down to the species level.
So Planet 3D is asking, in terms of coral pathogens, how do you determine which coral pathogens may be, or which pathogens may be tolerated by different corals? So obviously you guys can do that, right? I mean, in terms of identifying the different bacteria that could be impacting corals, but I guess, how do you determine what one is bad versus good?
So I guess I'm not exactly sure about the question, but let me answer what I think we're getting at, and let me know if it didn't cover it. So talking about bacterial pathogens.
Yeah.
There is a list of bacterial pathogens that we screen for. So step back for a moment and say that our first step with the bacterial data is simply to take every single sequence from your sample and ask, who is it? Which species is this? We identify each species, each type, as far as we can, whether that's the genus or the species level. Once we've done that, once we have identified everything in the sample, then I just have some code that goes through and it has a list of known pathogens. And this is based on published reports where people have identified this pathogen causes this disease. And so we identify, we detect pathogens in people's samples on that basis. We find things like, well, there's several. ARID is one of the most recent ones that we've been seeing a lot of. It's a very common, almost symbiont in nature. It occurs in about a third of corals in nature, and in low nutrient conditions, it doesn't cause any kind of problems for the corals. So it just hangs out there as kind of a neutral member of the community, not causing any problems. But when the coral is transported into high nutrient conditions, all of a sudden this bacterium, Aqua rickettsia roweri, it turns into a pathogen. It starts stealing nutrients from the host and leads to reduced growth and tissue wasting. Okay, anyway, my point there is just to say that's an example of a bacteria that was identified by researchers, presented in a peer-reviewed publication, and we've just screened for the name of that bacteria and found that it's in the sample. We have other sequences, other suspected coral pathogens. Some of them are based on peer-reviewed papers where I found somebody had done a study and we took those names or sequences and added them to our list. Others are based on our in-house work, such as this Archobacter species that you and I were talking about earlier. It hasn't been formally identified in a peer-reviewed publication, but boy, we've got a lot of good evidence for it. So I mean, broadly, we're using the DNA to ask who is this for each bacterial sequence, and then we're just comparing that list of names with a list of known bacterial pathogens. See if I hit any of what he was looking at in that rambling answer.
Yeah, no, I think so. He's also asking, what have you identified with regards to brown jelly and torches and hammers? Anything specific?
So I'm convinced that is a bacterial disease, and I know there's a school of thought out there that it's caused by ciliates. I am completely convinced that this is a bacterial disease caused by Archobacter, a particular type of Archobacter. It's not classified to the species level. We call it type 1103. This same sequence, we've found it associated with brown jelly disease on multiple continents now. And if you treat it with ciprofloxacin at a low dose, 0.125 milligrams per liter, you knock out that particular type of Archobacter and the other bacteria stick around. So it's really sensitive to cipro. So having seen it in both directions now, it's associated with the emergence of the disease, and if you knock it out with a specific antibiotic, the disease goes away along with that one type of bacteria. I'm quite convinced that that's the cause. What hasn't been done now is one of the important steps to really publish this thing would be to isolate the pure culture of the bug and deliberately infect a coral with it to demonstrate that that does cause brown jelly disease, and then re-isolate it from that coral. So if there's anybody with a lab out there wants an easy paper to write up, that would be a great, somebody really could sit down and finish the study on this thing to formally classify it as a pathogen. But in my own hands, I'm quite convinced bacterial disease caused by Archobacter 1103.
So you mentioned using an antibacteria, like I'm assuming you're also talking about it as a dip, right? You're dipping corals in this antibacterial agent to
yeah.
Correct?
Important question. That's exactly how I started out, for sure. I personally have not had success with dips of cipro to treat, that is to, address an existing infection of brown jelly disease. I do use a preventative cipro dip when I buy a bunch of Euphilia from a wholesaler, because the wholesaler Euphilia always come in with this bug on them, and I just do a preventative dip. I don't have good evidence that preventative dip is doing anything, because I don't waste half of the Euphilia as controls. They're too expensive. But yeah, in my own hands, I have not had success using a dip for a treatment. A colleague of mine has, Lex Inverts, guy we were talking about earlier. He's had some good success using a Cipro dip. The way that I use Cipro to treat brown jelly disease is actually as an in-tank treatment. That's the low dose that I just described. And the low dose is really critical. I'd be very, very cautious putting antibiotics in your tank. Yeah. I only did it because I had a tank that was almost only Euphilia, and they were all dying. So what do I have to lose? You know, so I tried it after some careful research and got lucky. It turns out. So well.
An in-tank treatment is something perhaps you might not recommend to somebody with a mixed reef tank.
Absolute last resort. If yeah, I mean, if you've got some sick Euphilia, your best bet would be to remove them to a
hospital tank.
Let's say a hospital tank rather than a quarantine tank, because the idea would be, you know, to treat them in that tank for a while. Yeah, that would be my preferred method, is remove the Euphilia into a separate tank and treat them with the Cipro. If you can't do that, or you don't care because it's only a Euphilia tank, then a low dose of Cipro in tank does seem to work.
All right, I'm going to address one more question from Planet 3D, and then that person has gotten a lot of airtime. So other folks, please ask your questions. But this person's asking a lot of good questions, so I'm going to throw them out there. You know, essentially the comment is that you guys have some excellent information, but do you also provide recommendations in terms of how to act on that data? You know, so if you come back with a certain coral pathogen, or if you come back and you're showing like the tank's got cyano and the person knows it got cyano, or are you coming up with recommendations, solutions that help the hobbyist solve these issues?
Right. That's an important part of the service, and it's also a major challenge. And I want to say that I've taken a stance on this that may be hurting me financially, but I think is the right way to go. And that is, I'm not going to give you advice unless we have evidence for it. So a lot of the stuff that we find in the tank, and let me take an example here of SCTLD. This is a coral disease, stony coral tissue loss disease. It's a Caribbean coral disease. It's a major problem right now in the Caribbean. We find 4 or 5 of the pathogens associated with this disease in aquariums. So here I'm telling you, you've found a pathogen in your tank, and you may want me to tell you what to do about that. But this is an ongoing area of research. You know, not only do I not know how to treat this disease in your tank, science doesn't know how to treat this disease in your tank. I don't want to be too critical about the aquarium hobby here, but maybe we can all agree that there are some players in the aquarium industry that are not so careful and feel free to prescribe you a bottle of something you could buy, even if there isn't really good evidence that that thing is going to work. If we don't have evidence that we have a treatment for it, I don't want to give you advice claiming to have that evidence. So as the company develops, you know, as our knowledge as a community grows, we're going to have more and more advice for you. When we first started, we had very little advice we could offer, but as we've gathered more and more data, we've learned more about these relationships, and now we can give you more advice. Let me give you a positive example here. I just told you I can't tell you how to cure a coral disease. Well, let me tell you something we can tell you how to address. When we first started looking at people's microbiomes, we saw they were all different. We started kind of classifying the patterns and seeing what microbiome was associated with what kind of an aquarium setup. One of the strongest signals, one of the strongest relationships we found is UV sterilizers. So people put UV sterilizers on their tanks with the idea of killing parasites and killing pathogens. I don't want to take a strong stand about whether they're effective for that purpose because I haven't done those experiments. But what I can tell you for sure is they kill the most abundant group of bacteria that's found on the natural coral reef, very, very specifically. So if you go out to the Great Barrier Reef, the most abundant group of bacteria is a group called Pelagibacteriaceae. The beginning of that, pelagi, refers to the pelagic zone. So these are free swimming bacteria plankton. They're truly floating around in the water rather than clinging to surfaces. And again, on a natural reef, it's the most abundant group. On your reports, it shows up as a big pink bar in your community composition. And we find that UV sterilizers specifically get rid of this group. So when we first started out with the company, and I'd get back a report, a profile that had no Pelagibacteriaceae, I didn't know what to tell you about it. Over time, we've learned it's very clear that the UV sterilizer is causing the absence of that group. So now I know what to tell you to do. If you want to increase that group, two things: turn off your UV sterilizer, and because it's a waterborne bacteria, get some water from either another clean tank that doesn't have a UV sterilizer, or some water from live sand or from the ocean itself. So yeah, my point in that story is to give you an example of this idea that as we gain more knowledge, we're able to tell you more about how to adjust the microbiome in your tank.
You know, it's an interesting point about the UV sterilizer because, you know, I use them in my systems, and you know, it's not really more for the. I'm not concerned about, you know, fish disease. I think that there's a lot of parasites, right, in terms of fish disease that UV sterilizer won't knock out. I guess there's some that it will. I use it for you know, like dinoflagellates. There's you know, certain strains of dinos that UV will you know, knock out, which I think is something that comes in very handy these days with all the different dino outbreaks out there. You know, can it kill the good guy versus the bad guy bacteria? You know, obviously you're saying that yeah, that can be the case, but it would be very interesting to kind of see some sort of research into that in terms of what that impact is on reef tanks. I mean, you know, I have very healthy corals in my systems, and I know other folks that use UV that have very healthy corals as well. But then there's also folks that don't use UV and have very healthy systems too. So it's an interesting question, and I would love to see some more in-depth data on that.
Yeah, I agree, and I definitely acknowledge that we don't have that experimental evidence that that the presence or absence of Pelagibacteriaceae hurts or helps corals. I guess I bring it up purely as an example of how we can give you advice about adjusting the microbial community in your tank. But you know, it's up to the end user whether they want to do that or not. You know, sometimes we may have a client, their major concern is just about the pathogens. Yeah. They're not trying to find the microbiome. Right. They just want to know about pathogens. Yeah. So
one more question from one of the viewers. Great Bear to Reef. Thanks, Paul. Yeah, everybody, if you want to hit that like button, please do so. We've got almost 100 people watching, but only about 31 likes there. So let's get those likes up, people. Scott Oliphant has a question. Are you worried about antibiotic resistance in the hobby over time? There seems to be a wide uptake of various antibiotic, even used simultaneously. We've walked this road with tetracycline.
Yeah, I think this is an excellent point, and I hope more people are thinking about this. I always try to clarify that I don't recommend prophylactic antibiotic use in a tank. You know, I think that's been one of the major problems in animal breeding and agriculture, has been the routine use of antibiotics just to enhance growth rates, because it, you know, reduces some infections along the way, but it also leads to widespread antibiotic resistance, which is a major problem. And I would hate to see this problem crop up in the aquarium hobby, especially if it was my advice that led to it. So please, yeah, let's not do, you know, preventative antibiotic treatments. That would be a surefire recipe for developing antibiotic resistance in the hobby. I see antibiotics in the hobby really, like I do with my own children, don't use it unless you have to. You know, don't just use antibiotics anytime there's a sniffle. Use it when there's a serious disease and there's no other way that you can treat this thing. And brown jelly disease is the only one that I have done that with. And I did it because I had evidence that it was bacterial, and I was specifically testing that theory, saying, okay, if it's bacterial, I should be able to knock it out.
Yeah, I mean, I've had a couple of folks on the show that do use ChemiClean as a preventative measure on their systems, and they report that you know, everything is you know, no impact, and it's just they've got clean, healthy systems. But you know, I don't mean to knock ChemiClean here, but it is an antibiotic, and it will, my understanding, it will take out the good bacteria as well as the bad bacteria that you're trying to eliminate that's causing certain issues, right?
Yeah, I haven't done a side-by-side study with ChemiClean myself, but I've read those posts where people have, I guess somebody sent it out to a sort of third-party lab and got confirmation that it really was an antibiotic. Is that right?
Yes, that's my understanding. Yeah.
So I mean, if that information is correct and it really is an antibiotic, then in principle it is affecting the community probably much more broadly than, you know, the targets you're going for. Of course, the dose matters too. You know, so without knowing the concentration, I guess I can't speculate about how much we would expect it to impact the aquarium. I will say we have lots of clients that run ChemiClean. You know, in the questionnaire, as you've seen, when you submit a sample, you kind of check off, here's the things that I do in my tank. And people mention they use ChemiClean and their tanks don't come back, you know, completely sterile or completely bizarre. It seems to me that whatever impact it is having on the microbial community must be not completely disastrous, because we don't see a huge impact. Certainly need to put that one on the list of controlled experiments to do, because it is, as an antibiotic, we have good reason to expect it's going to impact the community.
I mean, it's so easy to use, right? A red slime remover to put in your tank, and then bam, within a couple of days, it's gone. I mean, it's kind of like the easy way out. And you know, I've used it. It's always like caused another issue. So there's always been some sort of side effect that I've noticed whenever I've used you know, the ChemiClean to address cyano. Yeah. So I really do try to, my first means of rectifying the problem is always to use good old-fashioned elbow grease and to try to, like, you know, address the root cause of the issue. I think stuff like ChemiClean is more of a band-aid than you know, anything else.
I'm glad to hear you say that about elbow grease. Yeah, my recommendations for cyano generally involve a physical removal. You know, get the population out of there. Of course, you're not gonna remove every cell, but, you know, vacuum it, most of it out, right? Some good cleaning. B, I like to make sure the nutrients are detectable. So in my own tanks, I run into cyano problems when the nutrients become undetectable. So really thinking nitrates here. When I have undetectable nitrates, I run into cyano problems.
And the reason being, my understanding, is because the cyano is consuming, or bacteria are consuming, a lot more nitrates than they are phosphates, right? So when they finish consuming the nitrates, that happens first. Then they'll go on to the phosphates. So if you've got zero nitrates and you've got some phosphates, then potentially nitrate dosing could help, right? If you're raising up the nitrates.
So I have used nitrate dosing in those cases. These days I've stopped. I've had good success with nitrate dosing to combat cyano as part of sort of a multi-step process. But these days I don't do it anymore. I just feed more. My feeling is, you know, the ammonia, well, the nutrients in the food will be converted, as we all know, down the pathway and become nitrate. And there were a couple instances where I had some weird algal blooms from dosing nitrate itself that I haven't seen from just upping the food level. But I think I think either way is a valid way of, increasing the nitrate levels in your system. My understanding, part of the reason that that cyano do so well when there's low nitrates is that they can actually fix nitrogen that other elemental nitrogen that other groups don't have access to, and so they don't need the nitrate. They can sort of make their own. I think that's one of the factors that causes it. Anyway, so yeah, echoing what you said, elbow grease, clean them out, get some nutrients in there, and then the third thing, I have seen benefits in my own tanks. If I have a cyano bloom and I add some live sand, that seems to help. So there seems to be a microbial competition going on. Makes sense. Cyano are also bacteria. And it seems that by boosting the diversity along with the deep cleaning, that really helps cyano.
Yeah, that's interesting because we're going to get into in a little bit my results with you guys. I did a little experiment. Well, not an experiment. I did something, a major reboot on one of my systems, and I did a pre-test, a microbiome pre-test, and then I did a post-test. But I also did another test in terms of the Rubbermaid tub I was using to cook some rock for that transition. So we'll get into the details of that. But the interesting thing is that I have had cyano pop up after I rebooted this tank. And one of my theories is, and I don't want to make this more about me versus what we've been talking about in terms of the conversation, but one of my theories is that I had a lot of corals in that tank. A lot of that coral had encrusted the rock. I removed that rock. A lot of that encrusted coral got removed from the system. So less corals in the tank meant less corals to consume nitrates and phosphates, which means that there is a nutrient, excess nutrients in the tank, since one of the things that was removing those nutrients is now gone, right?
Yeah.
So I think that to me might be one reason why I'm seeing cyano in the newly rebooted tank is the subtraction of some corals.
I agree. Yeah, I've seen things happen like that with corals, where I've added a lot of soft corals to a system and seen the nutrient levels just completely different, right? Because you've added animals. And we've seen it with fish. You have a fish or two die and you take them out of the system, and now there's different nutrient levels. It's something that I think doesn't often get noted well enough is, well, I still have corals and I still have fish, but maybe just one less fish or one more fish may actually have been the tipping point.
I also added I also doubled my fish population, so that probably had something to do with it too.
Yeah.
It's just basic, stuff that makes a lot of common sense, I think. That's what we need.
A friend of mine who had one of the most beautiful coral tanks I've ever seen had a velvet outbreak in his tank. Obviously doesn't hurt the corals at all, but lost a bunch of fish. And so he ran his tank fallow for a while and struggled with nutrients as a result, because he didn't have all the fish around pooping, and lost a lot of corals just as a result of taking the fish out. So yeah, sometimes the solution or the cause of the problem is a pretty low-tech thing.
Yeah. A lot of people are saying that they've got elevated nitrates and phosphates and almost never see any cyano. So that seems to be a common observation here in the comments. So, all right, Andrew Beaumont, if I'm pronouncing that correctly, who I saw in the chat earlier, and he's a hobbyist and a biologist. He's a friend of yours out in Oregon.
Yep.
I'm going to make sure I pronounce Oregon correctly. I
think some would disagree with emphasizing the gun part, but let's keep the politics.
So how do you
out
pronounce
here, huh?
how do you pronounce the state?
Oregon.
Oregon.
Yeah, you said it right. I'm just
okay.
teasing.
Yeah, I've taken a lot of flack over the years because one of my favorite corals is the Oregon blue tort. So that's I always mispronounce it. All right, so Andy passed along some information to me that seemed to be very interesting, and I wanted to kind of throw it out there to the folks out viewing and have you kind of give your take in terms of what Andy has been seeing. But he was able to collect some evidence using your data that a strain of Cipro-sensitive bacteria, Archobacter, which you already mentioned earlier in the live stream, was likely responsible for the brown jelly disease in Ghanes. He also received some data from you last month that suggests the Archobacter may also be responsible for, or at least correlated with, RTN in Millipora. So according to Andrew, the exciting implications of this are that since we have been able to neutralize, I guess, the Arcobacter with in-tank treatments and dips with Cipro, we can potentially also use Cipro as a tool to treat corals that are starting to RTN. So you want to kind of get into that, Eli, in terms of what the implications are, what Andrew has found?
Yeah, so his samples, I mean, it's great having a local reefer friend that's also a biologist, because he's always collecting interesting samples or running interesting experiments for me. So I had detected this association between Arcobacter type 1103 and brown jelly disease in Euphyllia. I'd previously written up something on Reef to Reef about it, and we've now backed up that association in probably a dozen or more tanks, including a couple of tanks in Europe. So I mean, this thing is a pretty specific association between this one DNA sequence, type 1103 Arcobacter, and a widespread coral disease. Andy's samples have been really useful for extending that observation. It appears that this is a more general coral pathogen than we had originally suspected. So he's got good evidence from Goniopora that it was, in his diseased samples, it was hugely prevalent. In his control samples, it was not. And wiping, it was also successfully treated in Gonies with low dose of Cipro. Recently, he had some RTN and acros, and the same kind of evidence showed an association there. That is, it's one of the most abundant bacteria on the diseased corals, but it doesn't show up at all on the control corals. I don't know if he's done the treatment yet with the acros to say that it also, can successfully treat the infection in acros, but it's been successful in treating the other infected corals with the same pathogen. So if it's causative, if it really is causing the RTN and acros, then it suggests that we could treat it in the same way. Yeah, I think it's hugely hopeful. And boy, wouldn't it be nice if it turns out that many of our coral diseases are caused by one bug, and we can, focus in on that one bug.
What, as hobbyists, can we do? I'm assuming that these coral pathogens can be passed from one hobbyist to another by just simply, if you're trading frags, or if you're buying frags from, from somebody, an online vendor or what have you, and they have that coral pathogen, in their system. I'm assuming that it can get transferred that way by just kind of hitchhiking. Are we looking at a future potentially also, a very common thing is for acros is to dip them, in whatever it is, bare potassium chloride, what have you, and other dips out there? Are we also looking at a potential another kind of dip for these pathogens? Preventative dip.
Dipping a coral certainly presents less problems for the rest of your tank than an in-tank treatment, right? I would prefer, if we're going to use antibiotics widely in the hobby, if that's our future, I would prefer it was used as dips rather than as widespread use of in-tank treatments. But I have to say, I'm even hesitant to do a universal dipping regimen that I would apply to every coral with antibiotics. I personally don't do this now. I only do preventative dips for Euphyllia, and only if I've got them from wholesalers. When I get Euphyllia from other hobbyists, I don't dip them in antibiotics. There is some risk of causing antibiotic resistance, and also antibiotic treatments are not they're not zero stress. It imposes stress on the animal just to put it through an antibiotic treatment, quite aside from the impacts on the microbiome of the coral. The coral microbiome includes beneficial bacteria too. And so if you hit it with a high dose of antibiotics, there's a good chance you're wiping out good bugs along with bad bugs. So you're asking if that's our future. I'm not sure. It's not my practice now. Now you're talking about transmission of these bacteria. We know they can be transmitted for at least a couple of these coral diseases. They can be transmitted through the water. So they certainly could be transmitted on an infected coral frag. So I think it is a real concern. It is something we should be thinking about, but I am a bit hesitant to suggest that we should all jump to preventative antibiotic treatments. Let me suggest this as an alternative, and it's nothing new that I'm inventing. It's simply quarantining, coral quarantine rather than preventative antibiotic treatment. If we all quarantined our corals as carefully as many of us quarantine our fish, this would, if nothing else, give you that observation period where you can wait and see, look, that thing didn't RTN in the first 2 weeks or the first month. I can be more confident that it's pathogen free than if I didn't put it through that, 1 month quarantine.
What about corals that are stressed out? Amanda Meckley from ACI Aquaculture is asking this question. Is it possible that corals that are stressed are more susceptible, to these sorts of coral pathogens? A coral wholesaler, the corals that they're receiving are making a much longer journey versus, stuff that's purchased by hobbyists, within the United States, and those are going to be very stressed out.
Yeah, I think we can answer that absolutely yes. There's good publications that they've done with abiotic stressors such as temperature or light. If a coral is stressed because of its conditions, yes, it is more susceptible to bacterial infections. And that's another reason why I don't dip my corals quite as harshly as a lot of people dip their corals, because last thing I want to do is stress that coral out after it's just been through a shipping incident.
Lynn Reef Nerd is asking specifically for the Cipro, what would you recommend for the dip concentration for a Cipro, and for how long should the dip be? Any thoughts on that?
So this is one that I don't have experience with, simply because my attempts at dipping Euphyllia in Cipro were not successful. Let me say that another way. I've done a preventative dip in Cipro where I have no evidence it works because I haven't done the carefully controlled control versus treated, right? I've used it purely as kind of a Hail Mary, please don't die, my new coral. Andy, that you mentioned a moment ago, I know he's done some careful dip trials and has had good success with it. I know it's a higher concentration than the concentration that I use for the in-tank treatment. And of course, a shorter duration. Maybe we can get those details up in the notes or something about this later. I don't have the numbers at my fingertips.
Yeah, Andy, if you want to put something in,
but they exist, so we can pass them on to you. That was Lynn, is that right? I'll pass those details on to you.
Yeah, or Andy or whoever want to put it in the comments in the video. That's good too. Bert Minshew has a good question. Eli, have you tried to see the exact bacteria strands on RTN, STN for SPS? Is it Vibrio?
So I've done several swab trials on RTN, and I have found Vibrio types elevated in them. They were never Vibrio species that have been previously associated with diseases, so I didn't have an obvious smoking bullet, and they were also not completely clear results in terms of the infected ones had a lot of it, but the uninfected ones also had some of it. So my short answer here is I don't have a conclusive data set saying what is generally the bacteria associated with RTN. It's been a challenging thing to study because, as the listeners all realize, RTN moves fast. So, I mean, sometimes you observe RTN by finding out that it happened last night and your coral is gone. And at that point, I really, can't do useful experiments with it at that point. In Andy's recent samples that we talked about, where he had RTN in Acropora and Millipora, and we took the samples, they were not elevated in Vibrio. They had some Vibrio in them, but it wasn't a known pathogenic type and it wasn't especially elevated. What was really striking in those samples was the Archibacter. Broadly, I think there are lots of Vibrio that are associated with RTN and probably causing it. There are certainly some published papers where they've linked RTN in a specific coral with a specific Vibrio. I suspect it's not really one syndrome caused by one bacterium. It's probably multiple bacteria, all of which produce similar symptoms and cause this broad family that we call RTN.
So getting back to Euphilia, Great Bearded Reef is making a comment. A fellow club member and Euphilia guide uses Cipro, iodine, ChemiClean, and amino combo and swears by it for black jelly disease. If he sees tissue recession, he uses mostly iodine and aminos. That's quite a cocktail there, Paul.
Yeah. And so, if you want to knock out bacteria, that is one way to go, right? Is hit them with everything, knock out all the bacteria, and you'll probably get the one that you're after. I'd be much more comfortable using some cocktail like that as a dip than as an in-tank treatment.
Yeah, I don't know if that is a dip. God, I hope it's a dip.
I know there's I've lost his name right now, but there's another hobbyist that's written me with very, very detailed recipes similar to that. That's a cocktail of multiple antibiotics that he uses and claims good success for treating our.
What about? I'm kind of like jumping back and forth here between LPS and SPS in terms of coral pathogens, but the SPS, RTN, STN. Mike Paletta is a big proponent of using witch hazel. Thoughts?
So we haven't done an experiment on witch hazel, and I was just looking at. We have a couple of bottles of it in the lab. I was just looking at it today and thinking we've got to do that experiment. In the absence of an experiment, I'm always going to be tempted to say I don't know. The evidence I've seen on forums about witch hazel, it seems like it's usually focused on ciliates. Am I right?
I don't know.
Let me say I've at least seen some people saying, well, I did this witch hazel treatment and it removed this ciliate that I think is associated with coral disease. There are a few ciliates that have been published that have shown associations with coral disease in published studies. They are not the ciliates that people talk about in the forum posts about this subject, and we very rarely see these disease-associated ciliates in our samples. This makes me kind of scratch my head about what the witch hazel could be doing, but without an experiment, I can't disagree with someone else's observations, but I don't know the mechanism. I don't know what organism are we targeting. Is it effective at removing that organism? So really would like to see a controlled experiment done on this. It may be a very useful thing to add to your tank. I just don't have data.
Fair enough. Rob Upstate, thank you very much for the super chat. The comment is great chat. Fingers crossed, no coral monkey pox. Laugh out loud.
That's right.
So Eli, let's do a little transition. So you made a reference earlier about some test results from one of my tanks, and I thought it would be really cool to kind of go through some sample reports that I've gotten back from you guys on my 187 gallon tank that I rebooted, and I'll give some folks out there an idea what your reports look like and what the data looks like, and so just to give people some background in terms of what I wanted to test. A lot of you that follow my channel know that I did a major reboot on one of my tank systems, my 187 gallon tank, which was a mature reef tank for like 5 or 6 years, full of corals. It just everything was doing really well, and the corals were just growing like crazy, and it was kind of choking one another off. So it was getting to the point where I was getting some STN at the base of some of the corals because of the lack of flow, the lack of light, and it was tough to kind of tear down a beautiful reef tank, a very beautiful mature reef tank. It was probably the hardest thing I ever had to do in reef keeping, to be honest with you. So what my plan was, and there was like a ton of coral that had encrusted on the live rock. So probably like 95% of the live rock, the 125 pounds of Haitian live rock I had in that tank that were facing the light, about 95% of that was encrusted with coral. So it wasn't like I could just kind of clip these colonies off the rocks and start planting frags, because it was all full of encrusted coral, right? So the plan of attack was to swap out the rock, that Haitian live rock in the tank, with live rock that I picked up. It was dry rock, Carib Sea live rock that I was cooking in a 100-gallon Rubbermaid tub with the established tank water, and I did 10% weekly water changes with the water in that tank. And I had a heater, I had a couple of recirculating pumps. I had there was something like residual light hitting that tub, but no direct light over it. So I had it cooking for 6 months. I was dosing bacteria, the Microbacter MB7, or the Brightwell's Microbacter 7, that on a daily basis. So the goal was to try to retain the bacteria population in that system. It was a 187-gallon tank. I also have 2 frag tanks plumbed into that system with a lot of acros in those tanks, a 75-gallon and a 50-gallon frag tanks. I also put in a cryptic sump. So I essentially took out the Haitian live rock, chiseled off all the encrusted coral. So that's what I was talking about before in terms of losing a lot of the encrusted coral. And I put that rock into the cryptic sump. And what I did was I did a pre-test. Before I did all of that, before I swapped out the live rock, I did a pre-test, a microbiome pre-test, I'll call it. And then I also tested, after 6 months, the Rubbermaid tub to see what the microbiome looked like with that tub. And I did a third test about 2 weeks after that swap. I call it a post test. We haven't gotten the results back yet, I know, for that test. So I thought it would be an interesting experiment to kind of see how the biodiversity changed, in the system. I mean, does that sound like a valid experiment to you?
Sure, yeah. Sort of before and after comparisons within the same tank are always a good thing. Of course, formally to step back and just criticize or point out, limitations to experimental design. We don't have a control side by side where you didn't do all those manipulations to the tank. So we should have some level of skepticism about any change that we see. Can we attribute it to the manipulation you did, or is it simply a change over time, right? That would have occurred anyway. With that said, that's just my experimental design OCD kicking in. I think a before and after design like this is a very useful study.
And I also thought it would be useful to kind of see, does cooking rock like that, is that effective? Does that, kind of do what you intended to do, which is to, help bacteria colonize that rock?
Yeah.
So the first slide that I'm showing here is this the pre-test. So this is before I did the whole swap, and this is the diversity slide, and it actually gives a diversity score, and it says that my tank's diversity is among the lowest of the tanks we have tested. And on the scale of 0 to 500, it's rated at a 45. Do you want to just kind of explain, Eli, what that means?
Yeah. Yeah. So the diversity, we give you 2 numbers for diversity. One is the raw number. That is the number of types that we identified in your sample. And this is standardized for sequencing coverage. In other words, if one person's sample gets twice as much DNA sequences as another, we standardize for that, so you don't have to think about how that could influence it. The other number there is the percentile, right? Just converting that number to a percentile, so you can ask what fraction of tanks were lower than mine. And yeah, yours was quite low. Now, as you and I were discussing earlier, we didn't get as many DNA sequences as we would like for that batch of samples, and this will lead to an underestimate of your diversity.
Does
So based on my experience, based on the numbers we're seeing here, I believe your tank truly has a low diversity, but this absolute number is an underestimate of it. When we get the additional data, we'll give you kind of refined estimate. My prediction is it's still going to be low.
Is that, do you think, partly due to the fact that I'm using a UV sterilizer? I mean, you know, it's a 5 to 6 year old reef tank, but it does have a UV sterilizer on it. Does the UV have come into play at all in that data?
Well, you know, we tested your tank. I guess it was August of 2021. I was looking back at your old report. And you had a pretty reasonable diversity. It wasn't the highest ever, but it was nothing like this. It was a pretty reasonable diversity level last time. I could pull up that number. I know it was higher. And so my point here is there's been a change. Yeah. So previously, in 2021, we had your diversity score at the 29th percentile, and this recent sample that we took put your score at the first percentile. Wow. Right. 99% of tanks have a higher diversity score than this one. So that's quite a reduction, and that's before you did any experiment. So I think that's the first thing. That's the first thing I want to say about these. Sorry, I'm struggling with technology here as I try to switch back to the full screen Skype. That's the first thing I want to say about your results is that it appears there's been a change in the community in your tank that has nothing to do with the experiment you're discussing. It is simply a change over time.
Interesting.
I mentioned that the diversity score is an underestimate because of coverage, but I believe when we give you more data, you're going to come back and have the same conclusion. Diversity has declined.
So here's the diversity of the Rubbermaid tub, right? So this is interesting to me because this is, like I described, basically I just have the Carib Sea Life Rock, which is dry rock in there. I'm dosing bacteria on a daily basis, and I'm doing 10% weekly water changes, and that's it. There's no other equipment on there other than the heater and the pumps. Yet the diversity score is much higher. Yeah, this diversity score in your Rubbermaid tub is pretty comparable to the score in your tank about a year ago. It's again not the highest of any tank we've ever sampled, but it's I guess a percentile of 34 or something. You know, it's sort of towards the low end of the normal range. It's a diversity score that I would describe as moderately low or low but not extremely low, and it's much more we detected in your tank. Can you? So in terms of, did you succeed in establishing a diverse community in the Rubbermaid tub? Absolutely. That's way more diverse than you would get if you had just put some dry rock in that tub. Do you think the bacteria dosing is what is driving that? Because I was also dosing bacteria, the same bacteria, to the established system. But do you think the daily dosing of bacteria is possibly helping that? I'm pretty confident in saying that the diversity we're seeing in your Rubbermaid tub came from the water. We lost you there for a second, Eli. You. And I say that because those bacterial products. You cut out right. You paused for a moment. You cut out right when you were about to say the answer there. So I think what I heard you say, I'm not positive, is that you believe the diversity is a direct result of the MB7 bacteria dosing. Oh no. No, I think the opposite. I think it's the water. So I think what you were doing by moving water from your established tank into the Rubbermaid tank appears to have been very effective. At least some of those microbes in the Rubbermaid tank. Okay. I'm going to pause here and ask, video okay for you right now? Yeah. Okay. Yep. Yeah, and the reason I say that, a couple of reasons. One, I've tested that product in particular along with a few others, and there's only a handful of bacterial types in that product. Two, the bacterial types we're seeing in your tank look like the types that we see in a normal tank, not in a bacterial product. Gotcha. Your video's actually frozen, Eli. I could hear you fine, but your video's frozen. Oh.
Hopefully it's a network.
Now your audio is kind of cutting out. We'll pause for a second here. Hopefully get Eli back. Eli, can you hear me? Oh, we lost him. I'm going to try to reconnect. Let's try to reconnect there. Hang on, folks. Little internet situation going on Eli's end there. Darn internet. Maybe he should move to a different part of the house. I'm raining again. Probably raining again in Oregon. Yeah, Lynn, maybe. All right, I'm sure we'll get back with Eli in a second here. Go take a bathroom break, folks, if you want to here. Come on, come on. What's going on? Technology. All right, let's see. I think he's active now here. All right, let me let's give it a few minutes. I'm sure it's just the internet's blipping out on him on his end. He definitely has can be a tricky internet connection in his house, but yeah, maybe somebody else in the household there is using the internet, uploading some movies or something like that, downloading some movies. I don't know. Yeah, folks. So I like I had talked about, I had ordered a bunch of microbiome tests from these guys, and yeah, I just thought it would be interesting to kind of compare the results in terms of before I did this major change to the tank versus after the major change of the tank was done, because I think we read a lot about, there's a lot of anecdotal conversation going on out there in terms of people cooking live rock and Rubbermaid tubs or whatever, and so I think, to me, I wanted to be able to quantify it a little bit to see if that sort of practice would be beneficial to the dry rock to essentially make it live rock. And according to the results that we've seen so far, it looks like that is the case, that my theories are being validated. Try them again. Well, I hate to end this thing prematurely, but let's see here if I can. Maybe he's emailing or texting me. Let's see what's going on here. Amanda, I'm not going to sing a song. Huh? Anybody else got anything out there? Any experiences? Anybody else had any experience with using the AquaBiomics data that you want to talk about in the chat? Any other comments about the data from Eli? Let's see. Any questions for me? Well, let me at least show you some of the other data that I got back from these guys. Yeah, that's not a good idea, Bert. So let me show you guys the composition data that I got back from this. So this was the composition data of the established tank in terms of the all the different bacteria that were found in the results in this pre-test. And yeah, so Eli had mentioned that there was a lot of Vibrio in the sample, but I guess what he was saying earlier is that there was no evidence in terms of whether it was the good stuff or the bad stuff in terms of the Vibrio. There's different strains, is my understanding. I don't know. Maybe the power went out on there, and that happened to me once with Dr. Tim. Anybody remembers that? Anyway, so that was the pretest, and then this is the composition of the bacteria for the Rubbermaid tub. So yeah, Eli had been talking about this earlier, and it seems to show a much better diversity in terms of the bacteria population in the Rubbermaid tub. So it definitely seems like the swapping, the 10% weekly water changes that I've been doing with the established tank water have helped. And I did do this for 6 months, so that was an extended period of time. I know the folks that I was talking to about this whole process in terms of pulling out rock from an established reef tank and putting in dry rock, that those folks had actually cooked the dry rock for much less of a period than I did. I think in one instance it might have been 2 weeks, and they had a successful swap in terms of that, taking live rock out and putting dry rock in. So they only were soaking it for 2 weeks. Another person I think was doing it for a couple of months. I did it for 6 months. I wanted to be sure. All right. Keep trying Eli here. I don't know. I think there might be a... I'm going to see if there's an email I'm getting from him. All right. So, folks, I don't know. Maybe I'll have to get Eli back on at some point down the road. We got... We almost got through everything I wanted to talk about. And perhaps what the best thing to do for me is once I get back the post results on this little experiment, I'll just maybe do a YouTube video on that to kind of go through all the different results. So, yeah, that's probably the best plan of attack. One last shot for Eli. Thanks, Bert. I don't mind awkward silence, just an FYI. Patch in. Yeah. All right, folks, I think we're going to sign off. It's almost 8:30 anyway, Eastern Standard Time. I apologize for the technical difficulties. I'm sure there's a good reason why we lost Eli, but like I said, I'll do a follow-up video on this little experiment of mine and report back the full results of this microbiome pre, post, and Rubbermaid tub test. So, listen, I want to thank Eli for being on the show. I also want to thank the sponsors for the show, both Bulk Reef Supply and Ecotech Marine. And I also want to thank all you folks that were out there tuning in and participating in the chat. And finally, a big thank you to Paul, the moderator. I also want to let you know that all episodes of Rappin With ReefBum are now available as podcasts on Spotify, Apple Podcasts, Google Podcasts, Stitcher, and Amazon. My next Rappin With ReefBum live stream is going to be a good one. You're not going to want to miss this one. It will be on Thursday, August 18th at 7 p.m. Eastern Standard Time with both Dr. Sanjay Yoshi and Mike Paletta together on the same live stream. So I'm really, really excited about having these two legends of the hobby on together. Should be another great show. If you want to check out the full upcoming schedule of guests, then visit reefbum.com and check out the YouTube section. Until then, be safe, be well, and we will see you next time.